Miss Nikki here. We get to move on to part 2 of chapter 11, where we're going to talk about some more specifics involved with nervous tissue. Hopefully by now we understand the nervous system. So this is the master control or communication system within the body. We use the nervous system, specifically neurons, to communicate with one part of the body to another. It's always fast, it's always specific, and it can cause immediate responses. So if you remember, we've had two different examples. If we have the brain and it sends messages to and from via our spinal cord. So let's say, like in chapter 1, I'm hot. My skin receptors send a message to the spinal cord sends it up to the brain, and then you know that you're hot. And what are we going to do about it? Well, if we're hot, the brain's going to send a message out to either a muscle, or in this case, a gland, and that gland is going to cause us to release sweat. So the nervous system serves as a communication and controlling system within the body. We can see from this figure the sensory input. So the example I gave you before would be the skin, sensory input telling your brain you're hot, and the motor output would be your sweat glands producing sweat so that it can cool you off. So sensory information is information going to the brain, and motor information is information coming from the brain. So they'll use these words afferent. Think of afferent as ascending. Think of efferent as exiting. So in this example, this person is looking at a glass of water, how full the water is. It's sending that sensory information to the brain, and then there's an integration neuron, so a neuron between the sensory and the motor. That's basically the decision maker. And then we send a message out through motor output or efferent neuron. So you should know all of these terms. You should know that let me erase this. You should know that afferent is sensory, efferent is motor, and the interneuron is the connection in the brain between the two. So we have two major divisions of the nervous system. We have the central nervous system, which encompasses the brain and the spinal cord, and then we have the peripheral nervous system, peripheral on the outside. So this is the portion of the nervous system that sits outside of the central nervous system. So peripheral nervous system would be the spinal nerves that come out of your intervertebral foramen of your spinal cord. So then the peripheral nervous system we can break into spinal nerves and cranial nerves. Here's a great figure illustration from the book. You can see central nervous system, brain, and spinal cord. Anything that comes out of the intervertebral foramen, that nerve that's coming out, that's a spinal nerve. And then we have a few cranial nerves. They originate somewhere within the brain, and they usually go out to feed the face or the neck. We do have one that goes all the way visceral, which is the vagus nerve. And we'll talk about that when we get to cranial nerves. So the peripheral nervous system has what we call functional divisions, so based on what they do. So sensory, messages to the brain, motor, messages from the brain, and then there are subcategories within each of these. So both of these, if it says anything about sensory, that's going to the brain. If it doesn't, in this case somatic or autonomic, it's coming from the brain. So let's look at the figure. This figure does a great job, so let's break down chapter 12. We're going to talk about the brain and the spinal cord. Chapter 13, we're going to talk about the peripheral nervous system. And we said there are two functional divisions. We have the sensory division and we have the motor division. So remember, sensory is carrying messages to the brain and motor is carrying messages away from the brain. Under sensory, we do have two categories. We have somatic, this was on the last slide, and visceral. So we can have messages coming from the body, and we can have messages coming from the organs. For the motor division, we have somatic, and now we call it autonomic. So instead of visceral, we say autonomic. So somatic, S should mean skeletal muscles and involuntary. So these are all the neurons that control voluntary movement, impulses coming from your brain to your skeletal muscle. Autonomic is information going to your cardiac muscles, your smooth muscles, and your glands. And then we can further break down autonomic into sympathetic division and parasympathetic. So if a message is coming from your brain and it's telling something to speed up, that would be sympathetic division. If a message is coming from your brain that's saying conserve energy and slow down, that would be your parasympathetic division. On to the cell types. So neurons, these are the nerve cells. So what's in nervous tissue? Inside nervous tissue, we have neurons. Bundles of neurons together make nerves. A bunch of nerves makes nervous tissue. We also have something called neuroglial cells. So these are the supporting cells. So when we look at a microscope slide of nervous tissue, you'll see the dendrites, the soma, you'll see the axon, you'll see the little axon terminals, and then you'll have these little dots all over. And these dots are these supporting cells. Here we have the neuroglial cells of the central nervous system, so we should be thinking brain and spinal cord. Here's the astrocyte. You can see that it has its little perivascular feet wrapped around the capillary. Basically, it decides what's allowed into or out of this tissue. So I usually think of it kind of as the gate guard, right? It's patrolling the blood vessels and it decides what can get in and out. The ependymal cells, these guys are going to line the brain ventricles or cavities, and they're going to help produce cerebral spinal fluid. Then we have the oligodendrocyte, and you can see this here, little cyclops looking cell, and it's going to produce myelin, and that myelin is going to wrap around and insulate the axon so that the signal, action potential signals happen more quickly. Then last we have this microglial cell. These can basically act as macrophages. They can go around and destroy any pathogens that are present. There are only two cells that make up the neuroglial of the peripheral nervous system. We can see a satellite cell here that wraps around the cell body of the neuron, and then we can see the Schwann cells, which actually form the myelin. A few characteristics about neurons. Basically, they're large, highly specialized cells. They're long-lived, 100 years or more. They're amitotic with very few exceptions. Remember, amitotic means no division. So I'm sure we've heard people say you're born with all the brain cells you're going to have. It's not entirely true. We have seen some neurons grow, but for the most part, if you damage part of your brain, it's not going to be able to recover. They're also highly metabolic. They need lots and lots of glucose and oxygen. And again, why? Because they need those to make ATP. And your brain specifically likes glucose. Your brain does not want to have to use fat as an alternative energy source. It would rather have straight glucose. On the previous slide, there were a lot of words. Let's just make sure we can understand what they are in the figure. So this is the cell body or the soma or the perikaryon. I call it soma. There's the nucleus. This is where your DNA is located. And then there's the nucleolus. Remember, this is where that ribosomal RNA, transfer RNA, anything that you're going to need to make a protein or do translation comes from the nucleolus. The nasal bodies here, this is really your rough ER. And then we give you some new terms. We say ganglia and nuclei. So what they're saying is if I take a bundle of somas, so I have a soma and a soma and a soma, and I have a bundle of somas, and they're all going to have their little axons coming down, right? But if I have a bundle of somas in the central nervous system, so in the brain and spinal cord, we use the word nuclei. If I have a bundle of somas in the peripheral nervous system, we just use the word ganglia. So instead of saying a bundle of somas located in the central nervous system, I say that area is a nuclei, so you automatically know what I'm talking about. The dendrites are the arm-like regions or receptive regions. So basically a signal comes in, causes a local potential, then causes an action potential. The message travels down the axon and then causes some sort of neurotransmitter to be released at the axon terminals. Now we know about the axon hillocks, which is this area here where we turn a local potential into an action potential. We can see the axon, little pieces of it poking out all the way down to the axon terminals. So another term they'll use is to talk about axons. They'll talk about tracts versus nerves. So here are the somas, and we have a bundle of axons. So if I have a bundle of axons in the central nervous system, I call it a tract. If I have a bundle of axons in the peripheral nervous system, I will call it a nerve. So a few slides back, they mentioned the word retrograde and anterograde. This is the normal movement from a neuron. So we start at the soma, the message goes down the axon to the axon terminals. That's a normal movement. But there is something called retrograde. So instead of going down, things go back towards the soma. There are certain viruses and bacteria that can damage neural tissue by using this retrograde transport system within the neuron. So polio, rabies, herpes simplex virus, tetanus toxin, and basically you can have these viruses or bacterial toxins hide inside the soma of a neuron. So we've talked about myelin sheaths already. We talked about unmyelinated versus myelinated when we looked at nerve conduction. So remember, the unmyelinated was slow, steady signal, action potential generated. The myelin was the fast. So myelin is a protein lipid sheath wrapped around an axon. It's supposed to protect and insulate the electrical signal. It increases the speed of nerve impulse transmission. Remember the nodes of Ranvier, so there was a Schwann cell, a Schwann cell, Schwann cell, and this little area in between. You might remember saltatory conduction from last chapter, part 1. So there's two different categories. There's myelinated fibers and there's non-myelinated fibers. So fibers, I'm talking about nerves, right? Or neurons, which are bundles of nerves. Myelinated fibers look white. That lipid protein myelin gives off kind of a white shine. So we'll call them white matter. And then if there's non-myelinated fibers, they look more gray in color. So we have two things to look at here. This is gray matter and this is white matter. So white means that we have myelin and that it is a myelinated nerve fiber. You should also think fast. With the gray here, the gray is actually unmyelinated nerve fibers, and you should be thinking slow, steady signals. You can see there are many classifications for neurons. Here are 3 that we're going to look at. So there's a multipolar neuron. This has many dendrites, so lots of information coming in. All of that information converges on one output or one axon. Then we have one input, one output, shot. Then we have something for a quick reaction called a unipolar neuron. And basically, this is the receptive end, so you receive a message and it goes straight through to the axon terminals and it's going to release a neurotransmitter. You see how it doesn't even go through the cell body? This one went through the cell body. This doesn't even bother. And you can see lots of myelin. So this is for a quick, fast signal. We don't want to waste a lot of time. So even though we have these 3 major categories, you can see that there's a lot of variation within ththose categories. So this is a multipolar Purkinje cell of the cerebellum. Cerebellum has to do with coordination of movement. So this is lots of information coming in, you can see. So think of it as swinging a baseball bat. You need to coordinate all of your muscle action, your arms, your legs, your back. You need to swing the bat at a precise time in order to hit the ball, depending on how fast the ball is going. So you need a different type of neuron in order to handle that type of information and come up with one coordinated motion or movement. Let's kind of tie everything up together. This is summary. So we have sensory neurons. They're going to transmit impulses towards the central nervous system, right? So sensory is from the body to the brain. These are also called afferent, right? They're almost all unipolar. Remember we said this was for quick reaction. The cell bodies, so the somas, are located in the ganglia. This just is a collection of somas. And the somas are located in the peripheral nervous system. So imagine the somas here, and then we have the axon and the dendrite. So remember we have the dendrite, the message doesn't even go through the soma, and then it comes out the axon for that quick reaction or message. So all of the bundles for these neurons are located in the peripheral nervous system. For the motor neurons, and remember these are called efferent, the motor neurons carry impulses from the central nervous system. So from brain to effectors. It could be a gland or it could be a muscle, right? These are usually multipolar. We're going to have to send out lots of messages. So again, if we're swinging a bat, we're not just sending a message out to our forearm, it's to our forearm, our hand, our back, our leg. The cell bodies in this case are located in the central nervous system. Remember, the message is originating from the central nervous system, so it would make sense that the somas are located in the central nervous system. And then interneurons, sometimes called association neurons, these are the ones that lie between the motor and the sensory neurons, and basically they're going to shuttle signals around the central nervous system, the brain. They're going to make decisions. This is 99% of all of your neurons are interneurons in your brain that are relaying signals from sensory to motor. So another great figure from your book explaining all of this. Here's multipolar. We said motor neurons are multipolar neurons, and we said that the somas are located in the central nervous system. Well, this is the spinal cord, that is central nervous system. Then we said unipolar neurons are usually sensory neurons, or sensory neurons are usually unipolar. And we can see that the soma is located in the peripheral nervous system. It's outside of the brain and spinal cord. And then they give you an example of a bipolar neuron, where if we have messages from the eye, we want those messages only to go to a specific part of the brain. We don't need that visual message to go to our arm or to our leg. It just needs to go start at one point and go to the next point, which is in the brain. Before we talk about synapses, let's just talk about passing information. So if I have a neuron, we saw it talk to a muscle, right? Released a neurotransmitter, bound to the muscle, caused muscle contraction. And we called this area a neuromuscular junction. Well, you can also have a synapse. If I'm talking about connecting one neuron to another neuron in terms of information. So this neuron could release a neurotransmitter. It could be picked up by the dendrites of this neuron and cause this neuron to release its neurotransmitters. So this area right here where two neurons meet is called a synapse. And don't leave out that sometimes a neuron can talk to a gland, right? It could cause a gland to release sweat. Now they're just getting complicated. We have a presynaptic and a postsynaptic. Really easy. Here are two neurons. This is your synapse, right? Your site of synapse. So this is the presynaptic neuron, and this is the postsynaptic neuron. That's all they're trying to say. So here we have a because this is a site of synapse. So this is presynaptic, this neuron is postsynaptic. Here's another site of synapse. This one is pre, this one is post. Here's another site of synapse. This one is pre, this one is post. So let's look at all of these sites of synapse. So this is a synapse right here in this box. They're showing you the axon of, say, neuron number 1 is synapsing with the soma of neuron number 2. So we call it axosomatic. Now we have a third neuron over here. This is an axon, but now instead of touching the soma, it's actually synapsing with the dendrite, so we call it an axodendritic. And then we have an axoaxonic. So this is still all number 2, right? And here's yet another neuron, and instead of touching the soma or the dendrite, it's actually touching the axon. So we have 3 different sites of synapse in this figure. I like this picture because it shows you many presynaptic neurons touching the soma of one postsynaptic. So all of this material here, this darker color, this is all the soma of another neuron. So here you can see we've got hundreds of presynaptic neurons all converging on this one soma of a postsynaptic neuron. Now some of this is a repeat. We talk about neurotransmitters. The one we've seen before is acetylcholine, right? We know a synapse is when 2 neurons come together. And we know that at the axon terminal, you're going to release neurotransmitters. We've seen this with muscles. Well, the presynaptic neuron contains synaptic vesicles filled with a neurotransmitter. But you see that we're not just saying acetylcholine. So when we saw neuron to muscle, it was always acetylcholine. We're going to learn some other neurotransmitters. On the receptive end, the postsynaptic neuron has to have a receptor for that neurotransmitter. So if this neurotransmitter that's being released is acetylcholine, there had to be an acetylcholine receptor, right? We saw this with muscle. Here we have the presynaptic neuron, and here we have the postsynaptic neuron. You can see the synaptic vesicles. You can see that they have some sort of neurotransmitter, some sort of chemical, and then you can see that they have to have a receptor on the postsynaptic neuron side. So what are the chemical steps that are happening at these synapses? So the action potential arrives at the axon terminal of the presynaptic neuron. We have voltage calcium gates. Calcium is going to enter. It's going to cause the release of the neurotransmitter. The neurotransmitter is going to diffuse. It's going to bind receptors on the postsynaptic, and it's going to cause a graded potential in the postsynaptic neuron. It can either be excitatory or inhibitory. These neurotransmitters are going to eventually be degraded by enzymes. They can be taken up by astrocytes, or they can just diffuse away from the synaptic cleft. After the last slide, this one should be a lot easier to follow, right? So here's our action potential traveling on our presynaptic neuron. That action potential reaches the axon terminal. The calcium gates open, calcium comes in, causes the acetylcholine, or in this case any neurotransmitter, to be released from the axon terminal. So now we have our neurotransmitter released, and it binds to the receptor on the postsynaptic neuron. This is going to generate a local potential that could turn into an action potential in the second neuron, or the postsynaptic neuron. There is a little bit of a delay. It's called a synaptic delay. It's the time needed for the neurotransmitter to be released, diffuse across the synapse, and bind to the receptor. It takes milliseconds. So very quick, but there is a slight delay. So we're saying from the time this neuron releases its neurotransmitter, and by the time those neurotransmitters bind the receptor, that could take a little bit of time, milliseconds, and it's called a synaptic delay. In the presynaptic neuron, the receptors cause a graded potential if the neurotransmitter binds, and this is dependent on the amount of neurotransmitter that's released by the presynaptic neuron, the time that the neurotransmitter stays in that synaptic cleft. remember enzymes can break it down and remove it from the synaptic cleft. So depending on these effects, you can have two types of postsynaptic potentials. So again, this is the pre, this is the post. We're talking about what can the pre make the post do, basically. And there's two different categories here. There's excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So just keep in mind that this is happening in the second neuron or the postsynaptic neuron. So this example is showing you an excitatory synapse or an EPSP, excitatory postsynaptic potential. So remember that the pre had to cause the post to do this, right? So here is the resting membrane potential of the neuron at negative 70 millivolts. You have some sort of stimulus from the presynaptic neuron, and that causes the inside of the postsynaptic neuron to become more positive. So local depolarization of the postsynaptic membrane brings that neuron closer to its AP threshold. So if there's another stimulus, this neuron is very close to threshold. All it would take was a slight stimulus to cause this neuron to completely depolarize and have an action potential. This example is an inhibitory synapse. This is an inhibitory postsynaptic potential. So here we have our resting membrane potential. We have a stimulus. This is going to be from the presynaptic neuron. It either stimulates a negative gate to open so that negative items come into the postsynaptic neuron, or it causes the potassium gates to open and potassium leaves. Either way, it drives the neuron to become hyperpolarized. So it drops way below threshold. So basically this is inhibiting or stopping an action potential from being generated, or only a strong stimulus is going to cause this neuron to fire. Because we have to get from all the way down here up past threshold to generate an action potential. So you can think of the excitatory as priming the postsynaptic neuron, getting it ready to fire more quickly, and you can think of this inhibitory as stopping the neuron from firing or limiting the ability of the neuron to fire only if there's a really, really strong stimulus. So far, the only neurotransmitter we've seen is acetylcholine, but we have several different categories to look at. So there's over 50 different neurotransmitters, and we classify them by their chemical structure. So let's take a look at a few of those. So here's the first category that we're the most familiar with, and that's acetylcholine. It's released at the neuromuscular junction. We think about skeletal muscles when we think about acetylcholine. But we also have receptors. So now I'm talking about the receptors. This is the neurotransmitter. Remember we had the acetylcholine receptor, and what bound to the acetylcholine receptor was the neurotransmitter acetylcholine. So the receptors here, these are found in muscle. They can also be found in nerves. And there's two different types, muscarinic and nicotinic. So these receptors will bind acetylcholine, but these receptors are different. So they will cause different actions to occur depending on which type of receptor is present. So you can see muscarinic is present in cardiac and smooth, and nicotinic receptor is present in skeletal muscle. Do you see that they both bind acetylcholine, but they cause different actions? So the skeletal, if acetylcholine binds the nicotinic acetylcholine receptor, it causes excitation, right? Causes the skeletal muscle to contract. If acetylcholine binds a muscarinic receptor in cardiac or smooth, depending on the type of receptor, it can do two things. It can either inhibit or it can excite. It has two different choices, kind of like what we just looked at with excitatory inhibitory response in a postsynaptic neuron. This is why I could give you a block just for your skeletal muscles. So you go into surgery and I don't want you wiggling around on the table, so I can block your skeletal muscle excitation and contraction because I can block specifically nicotinic acetylcholine receptors. But I don't want your brain or your heart to shut down while you're in surgery. So I don't want to block your muscarinic acetylcholine receptors. So the second category is amino acid. These are several here listed, GABA included. These are involved in neuron signaling, learning, memory, and they cause inhibition in the brain. So remember when we talked about postsynaptic inhibition. So they cause that hyperpolarization, basically turn off neurons in the brain. So it's a neurotransmitter that causes inhibition or hyperpolarization in the postsynaptic neurons in the brain. So let's look at what alcohol does. So remember we said GABA, this was one of those amino acid neurotransmitters. It normally causes hyperpolarization or inhibition in the brain, right? Well, alcohol actually opens the channels that are usually signaled by GABA. This causes dopamine to be released, and that gives you the calm, euphoric, loss of inhibition, or relaxed feeling when consuming alcohol. Monoamines, this is the third group. You might also see it labeled biogenic amines. Either way is fine. Epinephrine, norepinephrine, dopamine, histamine, serotonin. These are involved in behavior and emotion. Sometimes we call them the feel-good neurotransmitters. They have something to do with regulating our mood, also our appetite. So how does cocaine affect these groups? Cocaine blocks dopamine from being taken up by the presynaptic, increasing the dopamine in the synaptic cleft, causing the postsynaptic to be stimulated again and again. So here's my pre. It releases dopamine. And here's my post. This dopamine binds to the receptors, and it causes this postsynaptic neuron to repeatedly generate an action potential and fire. So the postsynaptic neuron is stimulated again and again by the same dopamine. Usually this dopamine would get taken away or broken down by an enzyme. But the dopamine just sits there and basically pings the postsynaptic neuron again and again. This causes alertness. You feel really good. You have lots of energy. You have competence. Next group, I call them neuropeptides. I think your book just labels them as peptides. They're strings of amino acid. So remember, amino acid is just one. If you put the beads on a string, that's a peptide. These are peptide bonds. You have to go back to chapter 3. But these are involved in pain signaling, gut and brain interactions. So it has something to do with feeding behavior. So basically your stomach talking to your brain. So how does heroin or morphine play into these neuropeptides? So what happens is that the heroin or morphine actually binds the receptors on the neurons, the ones that normally bind neuropeptides. It disrupts the neural communication between the peptide and the neuron. So it's actually displacing the neuropeptides. It's pulling them off of the receptor and blocking or binding the receptor. These have something to do with the perception of pain. So your body can be in pain, and then you can take pain medication so that you don't feel the pain. The pain is still there, you're just not perceiving it. Your brain isn't aware of the pain. So the heroin or morphine binds to the receptor. It blocks the neural communication between the neuropeptide and the brain. And this gives you this euphoria feeling. You're in kind of a drowsy state. It causes the neurons to also make more receptors. So this is why heroin or morphine is highly addictive, because once you start taking the drug, this allows the neurons to make more receptors to bind more of the heroin or morphine, and then when you remove the drug, those receptors are still there, giving that addiction feeling, going, where's all the heroin or morphine that was here, right? The receptors are just sitting there waiting for more. 2 neurotransmitters, last 2 I want to talk about. One is nitric oxide. Causes smooth muscle to relax. This has something to do with male erections. So it allows blood to flow into the penis and maintain an erection. It's also responsible for peristalsis, which is alternating contractions in order to move food. Also has to do with kidney function. So you're going to talk more about nitric oxide and 2O2. I just want you to realize it's a chemical that acts as a neurotransmitter, causes some sort of reaction to happen either in a muscle or to another neuron. And then this last group, cannabinoids, I think they're fascinating because we didn't find out about them really until we started studying THC, or the drug that's in marijuana. So I have a slide coming up in just a second. But we make a group of neurotransmitters, and we call them cannabinoids. They help regulate our memory, our feeding behavior, and our motivation. So marijuana has THC in it, and this blocks our cannabinoid receptors. Well, we said cannabinoids normally regulate our memory and our thinking and our reflexes. Well, if THC blocks that, then you're going to have impaired coordination, right? So you don't have a memory, short-term memory is gone. You can't think really well. Your reflexes are slowed down. You might be really hungry because it has something to do with food behavior, right? So I wanted to talk about a specific monoamine called norepinephrine. So it's got the word epinephrine in it. You may think of this as adrenaline, right? It has a receptor that's an integral membrane protein. This norepinephrine basically can bind to this receptor that's in the membrane, and it can cause these various actions to occur. This is the important one, opening ligand gates. So I wanted to explain this second messenger system. So first we're going to go back and think norepinephrine, I said, is kind of like adrenaline. And then it uses this second messenger system, and it can open ligand gates. So let's say you've just ran a marathon, you're at the very end, you've used lots of ATP, your muscles are absolutely exhausted, your neurons don't have any more acetylcholine, you're wiped out, you're getting ready to pass out. All of a sudden somebody comes up and attacks you at the end. How do you get that energy? How do you get muscles to move if you're out of acetylcholine? So you can think of adrenaline, in this case I'm talking about norepinephrine, as a backdoor way to cause muscle action to occur. So basically the neurotransmitter norepinephrine binds to a receptor that's in the membrane, and that causes a string of events to occur, and that can activate an enzyme, it can create ATP, and it can open an ion channel. So what can norepinephrine do? It could open an ion channel which would cause sodium to flood into the muscle fiber cell, and it would allow contraction to continue even though you don't have acetylcholine. So basically it's a backdoor way to get a muscle fiber cell to fire if you need to. And adrenaline is only released if you're in sympathetic response, which is fight or flight, which is somebody's attacking me. It's my last ditched effort to get away from this person. So how do we turn off stimulation between two neurons or at a synapse? Well, the neurotransmitter can diffuse away, so it can diffuse out of the synaptic cleft. The synaptic knob, or axon terminal as we like to call it now, the axon terminal can reabsorb the neurotransmitters, the monoamines and the amino acids. So basically the presynaptic neuron can reabsorb the neurotransmitters that it released and use them again later. Then we also have enzymes that are going to degrade the neurotransmitters and break them apart in the synaptic cleft. So this is an example of acetylcholine esterase, which is a specific enzyme that breaks down acetylcholine. We also have to think about the fact that there can be modifications to the number of receptors on a neuron. So I briefly mentioned before that you can upregulate, you could make more receptors. Remember, receptors are proteins, and remember, neurons are cells. So the neurons can make the proteins that they need, and those proteins can go to the membrane and become receptors. Here's an example of a simple neuronal pool. So here we have our presynaptic neuron, and see how it comes out and actually synapses with several different neurons. So you have one presynaptic, and you have 7 postsynaptic neurons. So this is one message, and that message is spreading itself out to 7 different neurons. Here's an example of a divergent neuronal pool. Think about muscle action. So here is one neuron, and we know this would be the presynaptic neuron, right? And it tells these 2 neurons, which are the postsynaptic. These postsynaptic then tell 2 more neurons, so now this one becomes the pre, and these guys become the post. But basically think about, this is going to one neuromuscular junction, this is going to another neuromuscular junction, and this is going to another neuromuscular junction. So this could be the way that you send messages in order for your rectus femoris to contract. Remember, the entire muscle doesn't contract all at one time. You have several different neuromuscular junctions that are firing at different times. This is another divergent neuronal pool example, but what if we wanted to talk to many different muscles? So here's one message coming from the brain, and it is sending a message to this side and this, and then continuing on, and you can see 2 different outputs going in 2 different directions. Maybe I'm trying to swing a baseball bat. These messages are going to my arm muscles, and these are going to my leg muscles. But they all started with one original message coming from the brain. We also can have converging neuronal circuits. So think of maybe sensory. So here's one input coming in, the second, and the third, and they're all converging on one neuron. So let's say this is I smell fire, this is I see something, like fire in the background, some light, and this is I'm hearing the crackling that sounds like fire. All of those sensory messages are going to converge on one neuron so that you can decide what you're going to do about it. Here's a reverberating circuit. They think this might have something to do with repetition or short-term memory. You can see this neuron fires, then this one fires, then this one fires, then this one fires, and then it goes back and fires to this, which then goes back and reminds 2. So you're starting this circuit where you just continue to repeat the information. So think of trying to remember a telephone number. You just keep saying it to yourself over and over and over again. This is a very simplistic drawing that we think relates to higher mental activities. So they call it parallel after discharge circuit. So here we have one input, but it's targeting all of these other neurons, and then they kind of converge into one output. So again, you're taking in facts and information from multiple areas, and all of that information is coming together so that you can make one decision or one output. Let's look at some big picture questions to try to sum up this chapter. So why have the ability to excite and inhibit? So we had our presynaptic neuron, and we had our postsynaptic neuron, and we said it releases a neurotransmitter into this synapse, right? And then on the postsynaptic we have receptors, and those receptors will bind the neurotransmitter and generate either excitatory or inhibitory action in the postsynaptic neuron. So it'll either cause the neuron to fire, or it'll cause it to hyperpolarize so that it cannot fire as readily. So why would we have the ability to do that? Why wouldn't we want to just excite all of our neurons? Well, some neurons we want to inhibit. Probably the best example I can give you is if you walk into a home and somebody has 10 dogs. You walk in and you go, oh wow, I smell dog. Now you have to sit down and have a conversation with somebody. You do not want your brain to keep telling you, I smell dog, I smell dog, I smell dog, I smell dog. You would want to inhibit that postsynaptic neuron, stop it from firing so that it doesn't continue to tell you that you're smelling dog. Why does a neuron have the ability to detect more than one neurotransmitter? So again, I have this neuron, which is the pre, and let's say it releases acetylcholine. The only way the post will either be excited or inhibited is if I have an acetylcholine receptor. If I don't have an acetylcholine receptor, it doesn't matter that acetylcholine is released. It won't bind. So let's say over here I have a norepinephrine receptor. The acetylcholine is going to float by, but it's never going to bind. So why might we have more than one neurotransmitter to cause this postsynaptic neuron to do something? Well, because we might want to excite it or we might want to inhibit it. So we can also use different neurotransmitters in order to excite or inhibit the postsynaptic neuron. And then lastly, why would we have neural pools and circuits? Well, we have to transmit information in different ways, right? We're very complex. We may want to memorize something. We may want to do something short term, long term. We need to send messages out to all of our muscles, to a specific muscle. So we have a lot of actions that have to occur, so we need lots of ways or modes with which to get that information to the body. Well, that concludes part 2 of chapter 11. Make sure that you do the review questions. If you have any questions, please ask.